Yo, we about to break down DNA replication. Not fantasy, not fiction, just biology in action. High school science, but it's real advanced. So grab a seat, let's give DNA a second chance. Double helix, that's the SHRP of the strand. Deoxyribonucleic acid in demand. Made of nucleotides, got a phosphate group. Deoxyribose sugar and a nitrogenous troop. Bases pairing up A with T, G with C. Hydrogen bonds hold it down chemically. Before a cell divides, it's gotta make a copy. So replication starts and it's far from sloppy. DNA, we replicate it right. Semiconservative, one old, one new type. Helicase opens up the lane. Now we duplicate the code in the replication game. DNA, we replicate it right. Nucleotides match up day or night. Enzymes in control, precise and clean. This is molecular life behind the scene. Helicase unwinds, breaks hydrogen bonds. Creates a replication fork where work goes on. Single strand binding proteins keep it wide, so strands don't reattach or try to hide. Topoisomerase eases up the strain, relieves the supercoiling, reduces the pain. Now comes primase, it drops a short primer. RNA sequence, a starter not finer. DNA polymerase, main synthesis boss. Adds nucleotides, no time to floss. Always builds from 5 to 3. On the leading strand, it's smooth and free. DNA, we replicate it right. Semiconservative, one old, one new type. Helicase opens up the lane. Now we duplicate the code in the replication game. DNA, we replicate it right. Nucleotides match up day or night. Enzymes in control, precise and clean. This is molecular life behind the scene. But on the lagging strand, it's a different story. Built in fragments, no need for glory. Okazaki fragments made in bits, each with an RNA primer that fits. Polymerase builds, but it stops and starts. Ligase comes in with connecting arts. It seals up the gaps, makes the strand one piece. That's how a replication finds its piece. DNA polymerase checks for mistakes. Proofreads the strand, no retakes. Few errors slip, and if they do, repair enzymes, fix them like pros, it's true. Step 1, unwind helicase, got the code. Step 2, prime it, now replication mode. Step 3, elongate, polymerase go. Step 4, proofread, keep the error rate low. Step 5, ligase seals the strands. That's how DNA replication stands. DNA, we replicate it right. Semiconservative, one old, one new type. Built in the nucleus cell's control room. Copying genes like it's typed in a Zoom. DNA, we replicate it right. From G1 to S, we prep for the fight. Cell division next, but before it's seen, DNA replication keeps it clean.